Present. We all can see this, correct?
Yes.
Good.
Yes.
Okay.
Yeah.
All right. We talked about the plasma membrane in great detail on Monday, and today we're going to talk about what's inside the cell. Okay? So, the majority of the cell is cytoplasm, and the majority of cytoplasm is water. If you're ever asked a question on any exam and the choice is water, that's probably the answer, because the highest percentage of anything in your body is water. All right? So, what is the cytoplasm composed of? Well, it's composed of a gel-like solution called cytosol that's basically made up of water and soluble molecules like sugars and salts and protein. Inside the cytosol is also inclusions, pigments, fat droplets, crystals, holes, insoluble molecules. And also inside the cytoplasm is organelles, small organs that are the metabolic machinery of the cell. There either can be membranous organelles or non-membranous organelles, and we'll talk about each one of them, okay? Here are the organelles. The membranous organelles are mitochondria, endoplasmic reticulum, Golgi apparatus, and two big enzymes, peroxisomes and lysosomes. The non-membranous organelles are ribosomes, the cytoskeleton, and centrioles. So those are the organelles which we'll talk about in great detail. All right? The mitochondria. The mitochondria is what produces the energy for the cell. It produces ATP. Remember, I told you that when you talk about energy in the human body, you talk about ATP, adenosine triphosphate. So the mitochondria, because it produces the energy, is the power plant of all the cells. It is enclosed in a double membrane. The inner membrane is called the cristae because there are folds. And embedded in the cristae are proteins that play a role in cellular respiration. They use oxygen. The mitochondria contain their own DNA, their own RNA, and also ribosomes. There are two structures in the cell that contain DNA. Here's one of them, the mitochondria. The other one is the nucleus. So the two structures in the cell that contain DNA are mitochondria and DNA. Mitochondria resemble bacteria, and they are able to divide. And when they divide into another mitochondria, that's called fission. So here is the mitochondrion, that's singular, all right? And here are the cristae, the folds of the inner membrane. It has its own DNA. The genetic code is in your mitochondria, as well as the nucleus, all right? Ribosomes. Ribosomes are also organelles inside of the cytoplasm, but they are the site of protein synthesis. The function of the ribosomes is to make up protein, to make protein. You don't have to know the two types, that's not important. But when you hear the word ribosome, think you're making proteins. The endoplasmic reticulum. The endoplasmic reticulum is a series of parallel, interconnected valleys, and they're flattened membranous tubes. The endoplasmic reticulum is continuous with the outer nuclear membrane. There are two varieties of endoplasmic reticulum in the cytoplasm. One is the rough endoplasmic reticulum, and one is the smooth endoplasmic reticulum. The rough endoplasmic reticulum is called rough because it is studded with ribosomes, because those ribosomes will make proteins. And that's very important to understand. When the endoplasmic reticulum makes a protein, it has to be transferred for processing. And when it is transferred for processing, it is sent to the Golgi apparatus. We'll talk about the Golgi apparatus, but the Golgi apparatus' entire function is to process the proteins that are made by the rough endoplasmic reticulum. The smooth endoplasmic reticulum is continuous with the rough, and inside of the smooth are enzymes. These enzymes are important in making lipids. They combine lipids with protein and make lipid proteins. When you talk about lipid metabolism, you talk about cholesterol. The smooth endoplasmic reticulum makes steroids. The basis of all steroids is cholesterol. And when you talk about steroids in the human body, you talk about hormones. Most of the hormones are steroid-based. But the smooth endoplasmic reticulum is there to absorb, transport, and synthesize fats. It also will detoxify chemicals. It'll detoxify drugs. It'll detoxify pesticides. It is also important in converting glycogen to glucose so that you'll have some quick energy. And in the muscle cell, the smooth endoplasmic reticulum stores and releases calcium. Remember, I told you the most abundant mineral in your body is calcium. And here in the smooth endoplasmic reticulum, especially in muscle tissue, is where we store and release calcium so that you can move and your heart can beat. The Golgi apparatus. The Golgi apparatus's sole function is to package, sort, modify, and tag all of the proteins made by the ribosomes on the rough endoplasmic reticulum, the Golgi apparatus. And there it is. It's just a bag of bags, basically, to transport and package all the proteins made in the rough endoplasmic reticulum. All right? You don't have to know pathway A, B, and C. That's not important, and you'll never, ever use it. That's not important. Just know that it packages proteins. Peroxisomes. Peroxisomes are sacs of enzymes. And these enzymes detoxify substances. They are used in detoxifying free radicals. Free radicals are toxic, highly reactive molecules that are the byproducts of cellular metabolism and can cause havoc in cells if not detoxified. Actually, it can cause cancer. So you must get rid of free radicals. In the peroxisomes, there are two enzymes that have detoxifiers of free radicals. One of those enzymes is called oxidase. Oxidase uses oxygen to convert toxins to hydrogen peroxide, which is H2O2, which itself is toxic. However, the peroxisome also contains catalase, which converts hydrogen peroxide to harmless water. So the oxidase makes H2O2, and the catalase takes off an extra oxygen from hydrogen peroxide and turns it into water. Okay? So that's what you should know about peroxisomes. It neutralizes free radicals. Lysosomes. Lysosomes is another bag of enzymes, and these bag of enzymes are called hydrolases. Hydrolases. They are digestive enzymes. They digest bacteria, digest viruses. They break down and release glycogen. They break down and release calcium from bone. So they're hydrolases. Hydrolases are in lysosomes. Oxidases and catalases are in peroxisomes. Know those enzymes. A very rare disease called Tay-Sachs disease is a lysosomal enzyme disease that occurs basically in Central European Jews, and it causes severe seizures, mental retardation, blindness, and death before the age of 5. It's a genetic disease. The endomembrane system. This consists of all the membranous organelles that we already talked about. The endoplasmic reticulum, the Golgi apparatus, the secretory vesicles, and the lysosome. They produce, they degrade, they store proteins and fats. They degrade harmful substances. Those enzymes will degrade harmful substances, free radicals, bacteria, etc. And I'm sure you've seen this in your laboratory exercises. But let's talk about the structure on the skeleton of the cell. It's called the cytoskeleton, cell skeleton. It's an elaborate network of rods that run throughout the cytosol. It's the bones and ligaments and muscles of a cell. And there are 3 types. And you should know the difference between microfilaments, intermediate filaments, and microtubules. The microfilaments, the thinnest of all the elements. When we talk about muscles, there is a protein in muscle called actin, and that actin is a microfilament. It is the structure of the muscle itself. So I want you to know that microfilaments are the thinnest of all cytoskeletal elements, and an example would be actin, and actin is in skeletal muscle. And this is what it looks like. The actin looks like this, little beads, little beads. The smallest of all are microfilaments. Intermediate filaments, basically, you don't really have to know much about it, but it occurs in nerves. Basically, that's where it occurs. It occurs in nerves. Obviously, it is in the middle. It's intermediate. You go from micro to intermediate to microtubules. Microtubules are the largest of all the elements. Basically, microtubules are the motor proteins. They make things move. And that's what I want you to remember about microtubules. Largest and motor. Here they are, larger. They're larger than the actin. Motor proteins are all powered by energy. And that energy obviously is, again, ATP. There are other things inside these tubules, and these tubules inside are centrosomes and centrioles. The word centrosome basically means cell center. Okay? And basically, the centrosome is a organizing center for all of the microtubules. The things that are important is that the centrosome and the centrioles all have to do with movement. And the two things that they form is in big, bold letters, cilia. Cilia are hairs that are in your respiratory tract that get rid of dust, that get rid of bacteria by movement. And the other is flagella. Flagella is the tail of the sperm. Without this tail, that sperm cannot fertilize that egg. Here again, cilia and flagella. Hairs in the respiratory tract to get rid of dust. The tail of the sperm as the sperm swims up the fallopian tube to get into the egg. All motor, all motor. Another centriole or centrosome is called microvilli. Microvilli are finger-like projections that extend from the surface of the cell to increase the surface area. These microvilli are in your small intestine so that you can absorb the nutrients that you eat. Microvilli. Here you go. Cilia, respiratory cells. They sweep the mucus. They get rid of dust in your respiratory tract. They get rid of bacteria. Flagella, long extensions, a tail that propels the tail of the sperm so it can fertilize the egg. All right? We've beaten that to death. Microvilli, we talked about in the small intestine. Now let's talk about the nucleus. The nucleus is the largest organelle in the cell. It contains DNA. It contains the genetic library of all the mechanisms that will produce the cellular proteins. The majority of cells in your body have only one nucleus. Some cells have more than one. The only cell in your body that has no nucleus is a red blood cell, because a red blood cell doesn't need a nucleus. The red blood cell's only function is to transport gas. The structure of the nucleus are three main structures. The nuclear envelope, which is on the outside, the nucleoli, which is inside, and chromatin, where your chromosomes are. Here is what the nucleus looks like. And inside is a nucleolus. And chromatin is inside the nucleus also. The fluid that is inside the nucleus is basically called the nucleoplasm, the cytoplasm of the nucleus. And that's basically all you have to know about that. But in the nucleus is your DNA. And I also told you that the DNA is also in your mitochondria. Nucleoli are really not important. I'm not going to ask you any questions on nucleoli. I want to ask you questions on nucleus. Okay? But I'm going to ask you questions on chromatin. Chromatin are basically strands of DNA made up of proteins. Chromosomes are condensed chromatin. So when you say the word chromatin, you are talking about your genes, and that's where your chromosomes are. Your genes are in chromatin. Any questions before we talk about RNA and DNA? Okay. You still with me, guys, right?
Yes.
Good.
Yes.
Yeah.
Okay. I didn't like the way your book did DNA and RNA because it got into the weeds. So I stole this from a good friend of mine, and this explains RNA and DNA much better. So this is in your chapter lectures, and obviously will be on the YouTube channel sometime this afternoon. All right, DNA and RNA. DNA, what is it? Deoxyribonucleic acid, DNA. What is RNA? Ribonucleic acid. Where does DNA live? Well, it lives in the nucleus, and it also lives in the mitochondria. But the majority of DNA in your body is in the cellular nucleus. Why is DNA so important? Well, it's a nucleic acid that contains the genetic information used in the development and functioning of all living things and some viruses. You know, there are DNA viruses. You know, COVID-19 is a RNA virus, but there are also DNA viruses. DNA is the blueprint, the instructions, and the code for making proteins. Its code, the DNA codes are converted or changed into messages. And those messages are attached to messenger RNA for the ribosomes to read and make proteins. Remember, the nucleus has DNA. To make proteins, it has to be converted into messenger RNA. Messenger RNA will take this code to the ribosomes. The ribosomes in the rough endoplasmic reticulum will then make protein. Proteins do most of the hard work in keeping us alive. What are the parts of DNA? There are two strands known as the double helix. Here they are. The backbone has two parts. A sugar, deoxyribose, the D's, and a phosphate, the P's. Okay, so the backbone of DNA is deoxyribose, the sugar, and phosphate. Sort of like rungs on a ladder. If you look at the ladder, you will see that the rungs are basically amino acids. And in DNA, those bases are adenine, thymine, cytosine, and guanine. And I want you to look because A and T are always together. C and G are always together. That is DNA. A is always with T, C is always with G. How to remember nitrogen bonds? A bonds with T. Remember AT&T, the phone company. They're always together. C and G, remember half circles. So those are always together. A and T and C and G are always together. So here's a question. These are two examples of nucleic acid. Somebody give me an answer.
DNA and RNA?
DNA and RNA, you are correct. DNA holds the instructions for making
Proteins.
Proteins, correct. If 20% of DNA strand contains thymine, then
C?
yes, it also has 80% adenine. Remember, A and T go together and it's got to add up to 100%, right? All right. What type of sugar is found in DNA?
Deoxyribose.
Deoxyribose. The DNA's code is converted into blank so it can be sent to ribosomes to make the proteins.
B?
B.
mRNA, correct. All right, what are the parts of DNA? A sugar, here it is. A phosphate, here it is. A nitrogen base, here it is. All right? 3 parts of DNA, here it is. It forms a nucleotide. So a sugar, a phosphate, and a nitrogen base is known as a nucleotide. So let's practice here, okay? What are the complementary nitrogen bases in this sequence? What do you think AT and T would be? If A and T go together, what do you think AT and T would be?
TAA.
Yes, TAA. What about CGT? GCA. Everybody understand that?
Yes.
No, I don't get it.
No? Who said? Somebody said no.
Oh, wait, so the A
A
is
and
bonded
G
with
have
the
to go
T.
together. G and C
Oh, okay.
have to go together. So here's a here. TAT. It would be ATA. Who did I lose?
Okay, yeah, I get it. So the next one would be GCA.
Yeah, you got it. Okay. And this would be GAC. And here, this is an easy one. What's this one? AAA.
TTT.
Done. Okay. And then T. Everybody understand that? What
Yes.
did you do?
Yes.
What did we just do?
Create a DNA.
You made DNA. That's what you just did. Okay? Everybody got it? All right. mRNA, messenger RNA. DNA is too big and cannot leave the nucleus, so it must send messengers. mRNA, messenger RNA, is created by DNA in the nucleus. mRNA contains the messages from DNA and are sent to the ribosomes for them to read the instructions for making proteins. Does everybody understand that? mRNA does not look like DNA, and we're going to talk about the difference. So let's talk about RNA. Notice, RNA only has one strand. It doesn't have two. Okay? I want you to look here. Look at the nitrogen bases. Adenine, guanine, cytosine, and here's a new one. RNA has uracil. Which one is missing from DNA? There's no thymine here. Uracil has replaced thymine in RNA. But just like DNA, RNA has sugar, phosphate, and a nitrogen base. But RNA sugar is ribose. The phosphate is the same as DNA, but the nitrogen bases are different. Uracil has replaced thymine. So now instead of AT, you have AU. There it is. There is no thymine in RNA. It is uracil. Uracil.
So uracil replaces thymine?
Correct. That is correct. Okay? So instead of AT in RNA, it's AU. But it's always CG. Okay? Anybody else have any problems with that?
I have a question.
Go.
So since it's only one strand, will it still be A and U together?
Yes.
Okay.
Yes.
Thank you.
Yeah. Definitely. Definitely. There it is, uracil, a different nitrogen base. Okay? So here it is. A bonds with U instead of T, and C bonds with G. Did that answer your question?
Yes, thank you.
You're welcome. Good. Questions. What three things make up a nucleotide?
C.
C, sugar, phosphate, and a nitrogen base. Where is mRNA made?
The nucleus.
The
In the nucleus. You are correct. What type of sugar does RNA have?
Ribose.
Ribose. Which of the following nitrogen bases does RNA not have?
Thymine.
Thymine.
B.
Thymine. You are correct. You are correct. If a strand of DNA contains 40% cytosine, then
D.
D.
D. It also contains 60% guanine. Remember, C and G will always go together. All right? Okay. How does DNA tell the cell to make a specific protein? Well, there are two steps. Two steps. The first is called transcription. Transcription. The second is called translation. How does DNA tell the cell to make a specific kind of protein? Transcription. Transcription is the process of making messenger RNA from the DNA template. Transcription. Messenger RNA holds the recipe for making proteins. That's known as transcription. Transcription is when RNA is made from DNA. All right? How does transcription work? This is a great example. This is a great example. Have you ever been to court? There's a person typing what is said and is creating a court transcript, which is really a code, a shortened version. And later, the transcript is translated into all the words that were said for the record. So the shortened code is known as messenger RNA. Okay, now we're making RNA. Okay, here is the DNA code. Let's make RNA now. How are you going to make RNA from this?
Would it be, would you cancel out the TTs?
Say it again.
Would you cancel out the two T's?
No. Remember, this is DNA. This is DNA. I want to make messenger RNA now. I'm not making DNA. I'm making messenger RNA. This is DNA. This doesn't change. All of this is DNA. Now I want to make messenger RNA. So
Is it UAA?
very good. Everybody understand that?
Yes.
Yes.
Okay.
Yep.
Okay, so now go CGT.
GCA.
GCA.
Yeah, GCA. Okay. Now TAT.
AU.
AUA.
AUA. Okay. Remember, we're making RNA. We're making RNA. CGT would become GCA. CTG would become GAC. And what's AAA? UUU. And then ACG is UGC. You are making RNA. All right? This is called transcription. Everybody understand that? You're taking DNA's code and you're putting it on a one-strand RNA called messenger RNA. This is called transcription. There it is. All right, here's some questions. What does messenger RNA do?
B. Well, it carries instructions. Would it be B?
No.
A.
Carries instructions.
Hey.
A, it's A. Okay, it's A. What is transcription?
D.
D, the making of RNA. Let's do it again. Here's DNA sequencing. And now we're going to make RNA. So ACA would be
UC.
UG.
Well, UGU.
UGU. CGA.
GCU.
Yeah, good. You got it. You guys got it. What about TTA?
AAU.
AAU. CGG would be GCC. ATA.
UAU.
U.
UAU. GCG. What about ATC?
UAG.
UAG.
All right, we got it, right? There you go. What did you just do?
Me and RNA.
You made RNA from DNA. There it is. There it is. Okay. Now what? Yeah, exactly. Now what? Well, it has to be translated now. So the process in which mRNA attaches to the ribosome and a protein is made is called translation. The making of RNA, mRNA, is transcription, but to make a protein is called translation. You're now translating the genetic code. A codon. A codon is the 3-base code in DNA and RNA that we went through. We went through all of those 3-base codes, okay? There it is. We went through all of that. That's called a codon. An amino acid, compounds joined by peptide bonds to build protein. The basis of proteins is amino acids. There are 20 different amino acids. You don't have to know any of them, but and all of these combinations make different kinds of proteins. The ribosome. The ribosome reads messenger RNA recipes so it can make proteins. Remember, ribosome's function is to translate the code and make a protein. Transfer RNA. How does the protein travel through the cell? It travels through the cell on transfer RNA. That's how it transfers itself throughout the cell, okay? Anticodon is not important. Don't bother with the word anticodon. That's not important, okay? Where does translation occur?
A.
A.
No. Transcription occurs in the nucleus.
D.
D.
D, in the ribosome. What is made during translation?
Protein.
Protein.
Protein. What is another name for a polypeptide chain?
Carbohydrate.
Oh, no, definitely not carbohydrates. When you see the word peptide, the answer is protein. Protein. Well, we're not finished because I'm going to show you why this is important. A mutation is a permanent change in a cell's DNA, ranging from changes in a single base pair to deletions of large sections of chromosomes. You can get a mutation for various reasons: a virus, radiation, chemicals, or genetic errors in division of cells. Some mutations are harmful. Some do nothing, okay? Some mutations can cause cancer. Why is sunlight or a tanning bed a problem? Because it can cause skin cancer. And these are the 3 skin cancers that it'll cause. And when we talk about the skin, we will talk about these cancers, all right? And it can also cause lung disease and lung cancer also. This is a sickle cell. This is caused by the substitution of an amino acid in the polypeptide chain. Instead of having round disc-like cells, you have sickle cells. It's a horrible disease, but it can also prevent you from getting malaria because the malaria parasite can only live in these cells. It can't live in a sickle cell, okay? But sickle cell disease is a bad disease. You don't want that disease. And we can also make a fluorescent rabbit, if you'd like, a fluorescent jellyfish. The protein from this, the DNA code from a jellyfish, added to the DNA code to a rabbit, you can have a potential fluorescent bunny. No, it doesn't work, but it's interesting to see. Okay. DNA fingerprinting. How do you know if you're the father of your girlfriend's baby? Well, go on the Jerry Springer show and they'll do DNA fingerprinting for you and you'll see. All right. And you can clone. You can clone. Cloning is the process of which large numbers of identical DNA molecules are produced. This was the first animal closed. Her name is Dolly, and she is a sheep. She's now dead, but Dolly. There you go. All right. Let me stop recording.